students, good morning. How are you all? I hope you all are doing fine. So in today's class, we will study about the central dogma of molecular biology that we were discussing in the previous class. So in the previous class, what we were discussing? We were discussing about the RNA, properties of genetic material, right? And also we discussed that what is central dogma of molecular biology. So the central dogma of molecular biology means there is DNA, right? The DNA which is hereditary material. So as we all know, hereditary material should have the property of replication. That is, DNA which is hereditary material, okay, it should have the property of replication. That means to produce its copies or replica. Okay, so DNA, it will undergo transcription to produce RNA. Okay, then how the RNA is produced? It is produced by DNA, right? So the DNA undergoes transcription to produce RNA, and then RNA will have some information which will be taken from DNA. So this RNA, again, it will produce proteins, okay, it codes for proteins by the process called as translation. Okay, so this whole process, the way how the information from the DNA is transcribed or to the RNA, we call it as transcription, and how this RNA will, by the process of translation, how it will produce protein, this whole process we call it as central dogma of molecular biology. The whole molecular biology, it will rely on this process. Okay, so this process is proposed by Francis Crick. It states that the genetic information, it flows from DNA to RNA and RNA to protein. So in some viruses, this flow of information is reverse direction. That is why we call it as RNA to DNA, that is reverse transcription. So reverse meaning, so it is going in the opposite direction, reverse direction. So transcription should be from DNA to RNA, but here it is from RNA to DNA. Okay, so this process, again this process will be same, but the transcription is from RNA to DNA. This process we call it as reverse transcription, and we will study how it takes place. So before moving to the reverse transcription, we should know first what is replication and what is transcription. So we will study one by one. So DNA replication. As we have seen that DNA will produce its replica. Replica meaning its copies. So this process we call it as DNA replication. So replication is nothing but copying of the DNA from the parental strands. ನಮಗೆಲ್ಲರಿಗೂ ಗೊತ್ತಿರೋ ಹಾಗ our parents' DNA, right? So it will be inherited to us, right? The DNA or which is present in the chromosomes, yes, it will be inherited to us, right? So this process, that means the parental DNA has to make its copy. So ಅವ್ರ ಕಡೆಯಿಂದ ಪೇರೆಂಟ್ಸ್ ಕಡೆಯಿಂದ ಚಿಲ್ಡ್ರನಿಗೆ ಬರಬೇಕು ಅಂತಂದರೆ, ಸೊ ಪೇರೆಂಟ್ಸ್ ಡಿ ಎನ್ ಎ ಏನಿರಬೇಕು, ಸ್ವಲ್ಪ ಕಾಪೀಸ್ ಮಾಡಬೇಕದು. ಸೊ ಒಂದು ಸೆಲ್ ಡಿ ಎನ್ ಎ ನೆಕ್ಸ್ಟ್ ಸೆಲ್ಲಿಗೆ ಕೊಡತೈತಿ ಅಂತಂದ್ರೆ, ದ್ಯಾಟ್ ಮೀನ್ಸ್ ಇಟ್ ಹ್ಯಾಸ್ ಟು ಮೇಕ್ ಕಾಪೀಸ್ ಆ್ಯಂಡ್ ದೆನ್ ಇಟ್ ಶುಡ್ ಸೆಂಡ್ ಇಟ್ ಟು ನೆಕ್ಸ್ಟ್ ಸೆಲ್, ರೈಟ್? ಸೊ ಹಾಗೇನೆ ಪೇರೆಂಟ್ಸಿದ್ದು ಡಿ ಎನ್ ಎ ನೆಕ್ಸ್ಟ್ ಆಫ್ ಸ್ಪ್ರಿಂಗಿಗೆ ಹೋಗಬೇಕು ಅಂತಂದರೆ, ಇವನ್ ಇಟ್ ಶುಡ್ ಆಲ್ಸೋ ಮೇಕ್ ಇಟ್ಸ್ ಕಾಪೀಸ್. Right, so we have studied in the reproduction, so there will be millions of sperms, right? So these millions of sperms will have the DNA which is from parental side, that is paternal DNA. Paternal DNA andre from father, right? So that means the paternal cells, they should make a copy of DNA, many copies of DNA, so that all the million sperm should have the DNA copies. Okay, so for what it helps for to make the copies, the process of replication helps for the cells to make the copies of DNA, right? And even the same thing is happening in the egg, right? So even the egg will have the chromosomes from the mother, so even that means the mother cell should also make the copies of DNA, which will be in the form of which will be packed in the chromosomes, right? So this process, it is all possible because of the chromosomes, right? So this process is all possible because of the process of replication, right? So the replication is copying of the DNA from the parental DNA, okay? So parental DNA will make copies, okay, that we call it as replication. So Watson and Crick proposed the semi-conservative model of replication. It suggests that the parental DNA strands act as a template for the synthesis of new complementary strand. After replication, each DNA molecule would have one parental and one new strand. So Matthew and Messelson and Franklin Stahl proved the semi-conservative model. Okay, what happened is James Watson and Francis Crick, they proposed that the replication, it is in the semi-conservative model, okay, and this was again by the experiment, it was proven by Matthew Messelson and Francis Stahl. So we will see what is actually meant by semi-conservative model of the DNA replication. So before that, the scientists had different idea about how the DNA from the parents, it will be made copies, okay? So there were three models, that is semi-conservative model, conservative model and dispersive model. So they were not knowing actually how the DNA will make copies, whether in semi-conservative model, conservative model or dispersive model. So what do you mean by this semi-conservative model? So semi-conservative model is nothing but when the parental DNA, so the two strands, they will make copies. So this two strands will they will separate, and these two strands, the new strand, okay, new strand will be synthesized on these strands like this, okay? So that means semi-conservative, one DNA will be, one strand will be from parents and one strand will be newly synthesized strand. So in all the cases, okay, one strand will be newly synthesized and one strand will be from parents. So this is called as semi-conservative mode, okay? So you can see here, this is parental strand, okay? So it is the pink strands, two strands of the DNA. So what the parent will do, so it will undergo unwinding. Unwinding means the two strands will separate, okay, here, and the new strand will synthesize. The blue color, you can see the new strand will synthesize, and these will become parents when replication, and even that only happens here. So it will have the newly synthesized strand will be in one, and again it will have the inherited strand, okay? So again it will what it will do, synthesize its own strand. So here you can see, okay, in the next generation as well. So it is having pink and blue strand. So pink strand is from parental strand and new strand is newly synthesized strand, blue strand. So again when this also is producing the replica or copies, what it makes, it will also undergo unwinding. So one pink strand will go here and one blue strand will go here. So blue pink is nothing but just to give you an idea, okay, so how the strands will separate. And again the blue strand, again it will have its own newly synthesized strand, and pink along with pink strand, the newly synthesized strand will also make copies, right? So this process, the same thing is happening here also. So this kind of model we call it as semi-conservative model of DNA. So it is just that one strand will be newly synthesized strand and another strand will be from the inherited from parents. So the parental strand acts as a template and newly synthesized strand, that means it acts as a template model, and then newly synthesized strand will be complementary to it, okay? So like that the this is by the semi-conservative model. So what the conservative model of DNA says, so the parental strand, so it will not separate. One will be completely parental strand and one will be completely newly synthesized. And how this newly synthesized DNA will come, it was not explained properly. So this mode of replication, so it is not possible to occur only, right? So next comes the dispersive model. What the dispersive model says? Dispersive model says that both the strands will separate and they will be dispersed, like they will be in segments, and all the segments will distribute in these two, and even the newly synthesized strand will also be in the segments. Okay, so like that the next generation will also continue. Okay, so this is by dispersive model. So this is not there for your exam. Okay, so it is not even mentioned in your syllabus. Okay, but only you have to study is semi-conservative model, but you just have to know why they have done this experiment to know semi-conservative model is these were the three models that different scientists proposed. Okay, so different scientists believed, so the DNA may replicate in this model also, in this model also, in this model also. So through which model, like what is the proper model of replication of DNA, so that has to be proved by the experiment. Just by giving the theoretical theory, it is not enough. So that is why, to prove that the DNA it undergoes only semi-conservative mode of replication, they did different experiments. So here semi-conservative mode of the replication, one strand will be from parents, other strand will be from the new strand. Okay, so here completely one strand will be new only. So one strand will be completely from parents. Here all the two strands are from parents only. Here two strands are completely newly synthesized. Okay, so here you can see this this replication, it is not having any parental strand. So if it is not having any parental strand only, then how it will inherit parental characters? So this is not possible, right? So, and here in the dispersive model, what it is there, so all these the new strands and the parental strand, they are in the form of segments and new strands will be in between. So like that it is proposed. Okay, so which model is actually happening, taking place in the DNA? What is the exact model of replication? To prove it, there are experiments that were taken. Okay, so what are the experiments and what they have done to prove the semi-conservative model, we will see. So here we have studied that the Watson and Crick, they proposed the semi-conservative model of the replication. So they just suggested that the parental strands act as a template meaning, so it acts as one model, and in that the new new strand will synthesize. So they just suggested, and after replication each DNA molecule, so after the completion of replication, whatever each DNA molecule, the daughter strands, okay, so each DNA molecule will have one parental strand, okay, one parental strand and one newly synthesized strand. Okay, so Matthew and Michelson and Franklin Stahl in 1958. So this is very important. Nowadays they will ask even the year also. So you have to remember, in 1958 they proved the semi-conservative model. Who proved the semi-conservative model? Michelson and Stahl. Okay, so what is actually Michelson and Stahl experiment? How they proved the semi-conservative mode of DNA replication? Very interesting experiment. We will study it now. So what they did is, as we know, there were many different theories. So semi-conservative, conservative, dispersive. So these kind of models were also believed by different scientists. So they wanted to prove that it is only semi-conservative mode of DNA replication. So they did this experiment. So what is the experiment? So they prepared two culture media. So two culture media in the sense, in the two media, okay, media in the sense what it is, by the growing media, okay, so where the bacteria cells will be grown, okay, by providing the food that they require. So they prepared two culture media for E. coli. So Escherichia coli was grown in two different media, okay. So what they did, one preparation, it contains NH4Cl, 15 NH4Cl salt, that means N15 we call it as heavy isotope of N, okay. So this ammonium chloride salt, which is having the heavy isotope of N, that is N15, was incorporated in both the strands of bacterial DNA, and DNA become heavier. So what is happening here in this strand, the bacteria, so E. coli bacteria, its DNA was incorporated, that means N15, okay, ammonium chloride, in which N is, N15 is heavy isotope, it is incorporated, it is added, okay. So it will go and bind to the N molecule. So okay, so this N15, it is the heavy isotope, okay. So that means it is bound to the DNA. So this DNA, in which is the bacterial DNA in this media, will be what? It will be heavy, okay, because it is heavy isotope, right? So next what they did, so you just remember that N15, that is the nitrogen 15, okay, the isotope of nitrogen N15, it is present in this flask, okay. One bacterial culture, the medium will have the isotope N15, okay, ammonium chloride salt, in that N15 isotope will be there. So that will be put in this medium, and bacterial culture will be grown here, okay. So in the other stand, so in the other in the other flask, what they have put, the other preparation contains N14 salt. So N14 is nothing but N14 isotope. So N14 was incorporated in both the strands of DNA and become lighter. So this bacterial culture, the bacterial DNA will have what? This bacterial DNA will have N14, okay. So this become lighter. So the main character, okay, of the N14 and N15 is, wherever in the bacterial culture in this N15, since the N15 is heavy isotope, this DNA will be heavy, okay, which is present in this bacterial culture, okay. So since N14 is a lighter isotope, so when it is present in this bacterial culture, it will be lighter. The DNA in this bacteria will be lighter, the DNA in this bacteria will be heavier, okay. This you have to remember. And these two types of DNA can be separated by centrifugation in cesium chloride density gradient. So what they have used, very important, you have to remember, cesium chloride density gradient. So this technique they used, okay. So why they use this technique, how they determined the semi-conservative mode, that we will see now. So what they did, the cesium chloride density gradient, okay, it will help to determine the density of the DNA, okay. So after this they took, okay, so they took N15 here and N14 here. So the DNA here will have N15, which will be heavy. The DNA here will have N14, which will be lighter, okay. So what they did, they took E. coli cells from N15, medium and they transferred into N14 medium. So after one generation, that is after 20 minutes, they isolated and centrifuged the DNA. Its density was intermediate, that is hybrid between N15 and N14. So you just have to remember that heavier particles, what they will do? Heavy particles. So when you throw a ball, which will be, so cricket ball, if you think, if you throw it, so what it will do, it will soon fall in the ground, and for the gravitational force will be more acting on it. So if you take just a lighter plastic ball and throw, so it will be not too much attracted or gravitational force. That means the heavier particles will always settle down and lighter particles will always settle at the top, right? So it depends on the weight, okay, or the density, okay, that you can see. And that is why they took, what they did, they mixed this N14 and N15 medium, okay. So that means there will be transformation, as you have seen in the previous experiment, right? There will be transformation of the genetic material, right? So after one generation, so by the experiments we know that you have already studied that after 20 minutes the E. coli bacteria will go and they will produce the next generation. So that's why they did after one generation, that is after 20 minutes, they isolated and centrifuged the DNA, okay, from this mixed culture. It is there, no? So this mixed culture which will have, okay, this mixed culture, okay, this mixed culture which will have N15 and N14, okay. So both N15 and N14 will be present in this bacteria. So they have mixed, okay. So they just wanted to know how this N15 will be from the one bacteria, that is from one parent, and N14 will also be from one parent. So N15 will be heavy DNA, N14 will be heavy DNA. So when they both will mix, no, so what kind of next generation they will produce? On that basis you can easily find out whether it is semi-conservative method or what kind of mode of replication, okay. So after 20 minutes they isolated and centrifuged this DNA. So which DNA? This mixed from this mixed culture, okay. So the density was intermediate. So what they observed was the density, actually N15 will settle here because it is heavy, and N14 should settle here because it is lighter. But what they observed was it is not heavier or not lighter, it is here in between, it is intermediate or hybrid. So between N15 DNA and N14 DNA. So as you can see here, so N15 should be heavier, that is why it should settle near here, okay. So N14 it is lighter, okay. So that means it should be here, but here it is, see, this is hybrid, okay, N14 and N15, so it is in between. So but N14 it is lighter, so it should be here, okay. So what they did after 20 minutes, so N15 should be heavier, no? So but what they got intermediate, okay. So N15 and N14 it is there. So why it is getting intermediate? That means it is having, this DNA is having both heavier as well as lighter nitrogen, right, isotope. That means so they guess that one DNA strand will be, so both are heavy, both are intermediate, right. So that means one strand is heavier and one strand is lighter, that is why it is the density gradient showing it intermediate, okay. So that they got to know. And again by 40 minutes, second generation they wanted to observe, okay. So in the second generation, what they got, in the second generation they got hybrid also and they got lighter also, okay. So hybrid, why they got, so hybrid is nothing but N14 and N15 both together. But lighter, why they got, the lighter which is N14 is here. So because as you know, you have studied, so first what will happen, these two strands they will separate, okay. So when they will separate, no, so they will, in the first generation, that means they both will have one, okay, both the strands N14 and N15. Okay, so that's why it shows intermediate. Okay, so both the parents will have so the strands will have both one will be N14 and one will be N15. So it is neither heavy nor lighter, so it is intermediate. So but the next generation, what happens, again these two strands will also separate and these two strands will also get separated, right? So again what will happen, so one will be N14 and 2 will be intermediate. So after that's why after 40 minutes in the second generation, so there are two strands again which will be intermediate, right? One will have the heavy strand and one will have the lighter strand from the parents. Okay, so two strands will be having the the heavy strands and one will be newly synthesized. Okay, so and here the two strands will be lighter strands. So the heavier strands or the intermediate strands, okay, so having the N15 and as well as N14, both N15 and N14, that's why they'll be intermediate and they'll be formed here. And the lighter strands are 2, that is why here N14 will be seen, which will be a lighter strand. But can you see anywhere this heavier strand? No. So here heavier is not seen. Okay, only what you are getting first generation you got hybrid and second generation you got hybrid and light. You are not getting this heavy strand. So this proves that it is the semi-conservative method. That means from the parental generation. For example, this is father. Okay, so this is the first generation and again this is the second generation, right? So from here, so almost intermediate characters you are observing, and then from the next generation you are observing intermediate, hybrid as well as the lighter bands. So this was by the cesium chloride density gradient experiment. Okay, so you can see here, I feel you can see here, okay, how actually the experiment can be observed. Okay, E. coli which is having N15, E. coli which is having N14, their DNA. Okay, so when they both are mixed and when they are centrifuged and when they are run in the cesium chloride density gradient, the only heavier N15 will be like this. Okay, lighter N14 will be on the top, but they will you will be observing intermediate and lighter generations. Okay, so by this they call, see, if it is not semi-conservative, if it is conservative method, where one conservative in the sense one completely it will be having only parental DNA and completely newly synthesized DNA, that means it should show the heavier density here. So it is not showing anywhere the heavier density, so it is sure that it is not the DNA is not undergoing conservative method. And then the next which is that you just compare that experiment to this. So they proved that it is semi-conservative method because in the first generation they are observing the intermediate. Okay, in the first generation what they should observe? Intermediate, because they both are having both the parental as well as newly synthesized strands, right? But in the next generation you can observe newly synthesized strand also, so that's why they observed N14 band, lighter band, and intermediate also. You can compare it with this. So N15 as well as N14 both. So intermediate also they observed, right? So that is why by the experiment you just compare that experiment. Okay, so parental DNA will be present like this, so they have got intermediate bands in the first generation because they will have the newly synthesized, how it is having newly synthesized and parental strand, like that they'll be having N14 and N15, that is why they'll be showing intermediate band. In the second generation, what it is showing, both the heavy band also it is showing, intermediate band also it is showing, and lighter band also it is showing, right? So intermediate band means it will have the parental as well as newly synthesized strand, and heavy lighter band is nothing but they'll have only the lighter band, DNA, right? So that is the result of this experiment. So just compare, if it was conservative method of DNA, the replication, then in the first, when they'll conduct the density gradient, no, so all the one DNA it is showing completely conservative method, meaning what, the parental DNA will be conserved, so it will not share, or it will not what you call, okay, so it will not unwind, it will not undergo replication, so it will just form, it will just conserve its material, and only new synthesized strand will take by itself. So if it was the case, if you think the DNA is conservative model, then by the density gradient experiment in the first generation you should get the heavy metal, okay, so that is heavy band N15, okay, if it is N15 then you should get N15 which is heavy, okay, and only lighter band you should get because it is newly synthesized, N14 you should get, okay, but it is not showing like that, right? So it is not, never, nowhere it is showing the heavy band, right? So it can never be conservative model, right? But in the dispersive model, what is happening? So if it was dispersive model, in all the generation it should be what, intermediate only, it should not show the lighter band, okay, because in this method what is happening, in all the generation the DNA is it is spreading in the, or replicating in the fragments, okay, so even heavy strands and lighter strands, they should be in the fragments, and in each of the density gradient result you should get only the intermediate. But in the result it is not happening like that. In the experiment, what you are observing, in the first generation, yes, we observed intermediate, but in the second generation we observed intermediate also and lighter strand also. So this proves that DNA is not dispersive model, DNA is not conservative model, but it is semi-conservative model of replication. In this diagram also you can see semi-conservative model of the replication, okay, that is the reason, one, intermediate as well. So this is conservative model of the replication where only parental DNA will be here and newly conservative strand. Semi-conservative model is the DNA from the parental strand will be distributed equally, and dispersive model means it will be in the fragment form. So I hope you understood the experiment, so why they have done, okay, so they just so by this experiment it was very clear that the DNA is semi-conservative model only by looking at the result. So if it was conservative model, then you would never observe this intermediate bond, okay, so whichever you are observing, so it was clear that it is not the conservative model. So if it is dispersive model, so dispersive model is nothing but in every generation the DNA should have, it will be in the fragments, both the heavy and lighter strands will be in between. So you should observe only the intermediate bond in each generation, but in second bond it is showing intermediate, but in the second generation, so in the first generation it is showing intermediate, but in the second generation it was clear that it is not the dispersive model also, because the lighter bond is also seen. So only method that is accepted is semi-conservative method, where in the first generation, okay, you will get intermediate bond because of the presence of both heavy and lighter bonds equally, but in the next second generation both the lighter and intermediate bond was seen. So this proves the semi-conservative model of DNA replication. So this shows that in newly formed DNA, one strand is old, which is N15 type, and the other strand is new, that is N14 type. So this confirms the semi-conservative replication. And Taylor and colleagues, you should remember this also, in 1958 performed similar experiment on Vicia faba, which is faba beans, using radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosome. So what he did, he also performed some similar experiment. So what kind of experiment? It is not there for your syllabus, okay? It is not even there for your NEET and CET. So you just have to remember. So the same similar kind of experiment was done by Taylor and colleagues, and using radioactive thymidine, okay? So to detect the distribution of newly synthesized DNA in the chromosome. So how the newly synthesized DNA will be distributed in the chromosome, and it also provided that DNA in the chromosome also replicate semi-conservatively. So the chromosome, the DNA will also replicate semi-conservatively. It was also proven by his experiment, okay? So this is all for today. This experiment is very important because they will ask several twisted questions, okay? They'll give different situations. So what if N15, so as I told, why they did not consider the conservative model, so why they did not consider the dispersive model, okay? So every twist questions, many kind of twisted questions will be possible from this chapter, especially in your NEET as well as CET exam. So you just have to, if you know what is actually happening in this experiment, so that's why I tried to explain you with the different diagrams, so that you understand it very properly. So I also explained the different types of replication that were proposed by different scientists. So why they did this experiment, and what would happen if it was not semi-conservative method. So everything is explained, and I hope you understood this topic. So you just have to remember, what is central dogma of molecular biology, okay? So next class, so I hope you understood replication, and next class we'll study how the replication take place. Now we got to know that there is semi-conservative mode of replication, okay? So it is just that the experiment we have seen, but actual replication starts from tomorrow's class, where I'll explain the process of replication, the machinery, and we will study it tomorrow. So if you like the video, please click on the like button and share it to your friends, whoever is in need, and thank you so much.